Evening, everyone. Welcome to the fourth International Conference of RX Doctors, SYCCON Season 4, Episode 80, Organizing Partners SmartTech Academy, powered by OECC, EWTI, RHTC Canada. Today, we have Journal Club presentations on the 8th day of SYCCON. Now, I am inviting the judging panel expert Dr. Erin Nathania, PharmD, Project Associate 1 and Research Officer, CMC Vellore. We welcome you, ma'am.
Thank you so much.
Without further ado, let's start the event. The first participant today is N.L. Swati, Sri Venkateswara College of Pharmacy, slot number SYCCON RXS4 8002.
Hope my screen is visible to everyone.
Yeah, it's
Is
visible.
this okay? So let me come up to this right point. So as a general presentation, we need to give valuable insights. So that's why we are thinking like I have thought, what if I combine two articles which can give a valuable references rather than a single one. So that's why I have clubbed two research articles which says about how the metabolic disease or disorders transfer from parents to children's where there doesn't change any DNA. In some changes like non-communicable diseases or metabolic disorder where the DNA is not transferred from parents to children's, but we have seen like diabetes if the parent is having child is having, so that's why I have focused on this epic. So now the focus will be on epigenetics. It is a
Hello.
type of study which says that yeah,
Uh, could you actually zoom it or put it on slideshow?
if I
Uh,
put it on slideshow ma'am, it gets freezed, so that's why I'm saying like this.
oh, okay. Is it possible to zoom?
Yeah, I'll do it.
Okay. And if possible, you can hide the outline of the PPT also.
Yeah, is it
Outline
visible
slides.
ma'am?
Yes, yeah.
Okay, so well, I am trying to study what is the type of mechanism under the transfer of non-communicable diseases and metabolic disorder where the DNA doesn't get transferred from parent to children. I found an interesting subject called as epigenetics. This study inherits all the information where the inheritable and reversible changes affect the gene function to parents also. So now these two papers are going to focus on epigenetics, where they will say how they have been transferred from parents to children's. And so mainly the epigenetics deals with three types of modifications. First one is histone modifications, like a combination of proteins, like we have histone octamers, all this stuff, and DNA methylation, where the most of the part of this paper will be covered only under DNA methylation, and non-coding RNA slices. So I have kept a small video regarding DNA methylation. So let's say our DNA is yeah. So our DNA normally consists methyl group, which is usually attached to the DNA. So now let's say what happens if a person is having this changes. Something like the methyl group will be inactivated by binding by inactive subscriptions. Like as you can see, this is the yellow one is an unnamed or unknown substance which get binds to the methyl group. So this makes the compound inactive and makes the DNA damage. Let's say the DNA doesn't damage, but it is not a good DNA as well. So this is the process of methylation where the inactive group of inactivity of methyl is seen. So now for my second question, how does it transfer from parents to children? As you can see, one of this image where a rat is taking all of these junk foods like fatty acid and anti-saturated fatty acid foods high, and you can see the tissue morbidities which can be seen from opaque loosening and straightening. These are tissue changes happens when a person is taking the unsaturated fatty foods mostly. So some it will lead to his DNA which is inactivation, and this methyl group is getting same like that to the patient, to the children's itself. So this is not a damaged DNA, but a not working DNA. So this is what the epigenetics says. And so this is the trial rate where they have combined a protein called thyroidoxin interacting protein. So the major of this study was conducted on this protein itself, where the DNA methylation and this protein combines as a fuel for the DNA modifications or DNA inactivations. So this study mainly focused on glycemic changes and weight loss interventions overall, because the rationale of choosing this protein is it's a like dimorphic protein. It coincidentally involves in the weight loss and as well as in the glycemic changes. And let me say, like, what happens if there is an hypomethylation? What happens if there is a hypermethylation? So if hypermethylation, there it might leads to hypoglycemic attacks. Hyper means hypoglycemic attack will be like inversely proportional. Hypermethylation means hypoglycemia. Hypomethylation means hyperglycemia. So that's why these two are rationally for combined and mainly focused on type 2 diabetes and obesity, as this protein interacts with the weight loss and as well as um, glycemic changes. So how does the study was done is like, it was a 2-year randomized control trial where 639 adults were taken, and their um, protein, thyroidoxin protein, was monitored using DNA M level, which is a sophisticated test conducted over there. So this was a statistical analysis of the study. So now they have, as it's a randomized control trial, there was two models, model 1 and model 2, where the model 1 doesn't any we didn't do they didn't do any changes. They just took whatever the diet they have given, what they are taking, and they modified, and they checked the DNA M level and hypoglycemic level. And for the model 2, they have given a perfect diet which increases the methyl group inactivations. So that's why the HbA1c reduces and the obesity. So they have prepared a diet in such a way for the model 2. And this was the interaction between the DNA M level and takes an epi. T1 here, T1 here says that uh, the value of protein at the first stage, second stage, and the third stage upon giving the diet. So this was the this first graph is an co interaction between the fasting glucose and the thyroxine protein, and baseline HbA1c with the thyroxine protein, and homostatic assessment model of insulin resistance. So with this graph, they have come to an form end like there was an initial relationship between thyroid reduction protein and DNA M level as well. And this is the model's fasting blood glucose levels. Let's say how the model 1 has a 8.74, and the model 2 which is of 8.74. The 4 points has been increased by model 2 itself. So they have added more methyl group. So that's why the patient is having hyperglycemia. Like that, they have come to an endpoint. And these are all the p significance value to say that the results has been valid enough, like that.
Your
And
time,
total, yeah.
your time is up. 5 minutes completed.
Okay.
Over to
You
you,
can,
ma'am.
yeah, I just want her to, quickly finish the conclusion.
Okay, ma'am.
Okay, so according to the, we have found that there is a study which is epigenetics, which says that irreversibility and transfer of DNA, how it happens in the case of non-communicable diseases and metabolic disorders. And there is another paper which combines the epigenetics with the nutritional supplements, which was taken regularly during a lifestyle of a person. So this too can make a significant impact on transferring of non-communicable diseases and also type 2 diabetes mellitus. Ma'am. This was a conclusion of these two studies.
Okay, I do have few questions to ask.
Yeah, ma'am.
Um, on what population was the RCT done?
Ma'am, it was totally done on Asian and African peoples.
Okay. So, how did they? What was the method of randomization to study?
Ma'am, it wasn't like a single blind, ma'am. Only the principal investigator know the patient in the either in the model 1 or model 2. That's it, ma'am. A single blind randomized control trial.
Do you think this is reversible or irreversible?
Ma'am, the paper says that if you can correct your nutritional supplements at lifestyle, then it's a reversible change. But there isn't any appropriate reference for this conclusion, ma'am.
Okay. Um, so, what type of diabetes is mostly prevalent under epigenetics?
Ma'am, they have taken only a consideration of type 2 diabetes mellitus, and the
Okay.
study was going in various diabetic populations. Now they have been doing under gestational diabetes and as well as type 1, ma'am.
Okay. Okay. So, what are the other types of diseases due to epigenetic changes?
No,
Any
ma'am.
other example other than
Epigenetics is an
diabetes?
area which studies the transfer of this non-communicable diseases. Epigenetics is an area like pharmacology subject like that, ma'am.
Yeah. What are the other types of diseases that could be caused due to epigenetic changes?
Hmm. The other diseases which also includes rheumatoid arthritis, which is an autoimmune disease. But there is an research study says that if there is an in a family history of 10 to 15 generations, if a person is having rheumatoid arthritis, then the fifth generation might have the susceptibility of rheumatoid arthritis. One. So now they are currently doing in rheumatoid arthritis, and the other one is most of the lifestyle modification diseases, ma'am. HTN, hypertension, and dyslipidemia, and obesity, ma'am.
Okay. What could be the cost of providing such kind of treatment, like general? Have you gone through that?
No, ma'am. The cost is like typically a highly productive thing, ma'am. The DNA M level is needs to be measured for a person to say whether he is under epigenetic study or not. But this DNA M level is certainly a highly sophisticated test. So it's truly impossible for a normal person or a middle class person, ma'am.
Okay, okay. Are there any other studies from our Western data?
Yeah, we do have, but there is like a less amount of research papers and qualitatively sounded, but we do have a vast amount of literature surveys now besides this research paper.
Okay. So how do they measure the outcome for the study?
Ma'am, till now from what I have seen in the research papers is like using a comparative model or an ANOVA regression model or a multiple regression analysis, ma'am. Using this three statistic they have compared the efficacies of this methylation. And now for now, histone modification is a complex one where we couldn't even see the laboratory values also. So now the non-coding RNA and the DNA methylation. Only these two epigenetic changes were observed currently, ma'am.
Okay, okay. Thank you so much. Thank you. We
Thank you,
move on
ma'am.
to the next.
The next participant is Joanne Marilyn Wilson and Janavi from Krupanidhi College of Pharmacy. Slot number SYCCON RX S4 8006.
Good evening, everyone. I hope you can see the screen. Is it visible?
No, it's not visible.
It's not visible. One sec. Is it visible now, ma'am?
Yeah, it's visible.
Okay.
The time starts.
Good evening, everyone. I am Joanne Marilyn Wilson, and I'm from Kripanidhi College. So today I'll be doing my journal club presentation on the effect of ivermectin on time to resolution of symptoms among adults with mild COVID-19, a randomized clinical trial. So it's taken from the journal Journal of American Medical Association, published 2021. So a brief introduction is that so therapeutic approaches are needed to improve outcomes in patients with COVID-19. Ivermectin, a widely used drug with a favorable safety profile, is thought to act at different protein binding sites to reduce viral replication. Because of evidence of activity against SARS-CoV-2 in vitro and animals model, ivermectin has attracted interest in the global scientific community and among policymakers. Several countries have included ivermectin in their treatment guidelines, leading to a surge in the demand for the medication by the general population and even alleged distribution of veterinary formulations. However, clinical trials are needed to determine the effects of ivermectin on COVID-19 in the clinical setting. Viral replication may be particularly active early in the course of COVID-19, and experimental studies have shown antiviral activity of ivermectin in early stages of other infections. So I've used the CONSO 2010 checklist, and the topics here are title, abstract, introduction, methods, sample size, randomization, allocation, binding, statistical methods, and finally the results. And the page number, according to the article, is given over here. But because of the time limit, today I'll be mainly focusing on the introduction, study overview, main findings, appraisal, clinical relevance, discussion points, and conclusion of the study. So this study is identified as a randomized trial. And the background is ivermectin is widely prescribed as a potential treatment of COVID-19 despite uncertainty about its clinical benefit. Is double-blinded study, randomized trial conducted at a single site in Cali, Colombia. Potential study participants were identified by simple random sampling from the state's health department electronic database of patients with symptomatic laboratory confirmed COVID-19 during the study period. A total of 476 adult patients with mild disease and symptoms for 7 days or fewer were enrolled between July 15 and November 30, 2020, and followed up through December 21st, 2020. Result among 400 patients who were randomized in the primary analysis population completed a trial. The median time to resolution of symptoms were 10 days in the ivermectin group compared with 12 days in the placebo. By day 21, 82% in the ivermectin group and 79% in the placebo group had resolved symptoms. The most common solicited adverse event was headache, reported by 104 patients given ivermectin and 111 who received placebo. The most common serious adverse event was multi-organ failure, occurring in 4 patients, 2 in each group. So over here, the potential study participants were identified and selected by simple random sampling from the state's database. Adult men and non-pregnant or breastfeeding women were eligible in their symptoms began within the previous 7 days, and they had mild disease, defined as being at home or hospitalized, but not receiving high flow nasal oxygen or mechanical ventilation. Settings and locations where the data were collected. This double-blind randomized trial of ivermectin versus placebo was conducted from July 15 to December 21, 2020 by Centro de Estudios en Infectología Pediatricia in Cali. Study candidates were identified from the State Health Department electronic database. The outcomes. So the primary outcome was the time from randomization to complete resolution of symptoms within the 21-day follow-up period. The 8-category ordinal scale used in the study has been used in different 19 therapeutic trials and is recommended by the World Health Organization. It consists of the following categories: 0, no clinical evidence of infection; 1, not hospitalized and no limitation of activities; 2, no, not hospitalized with limitation of activities, home oxygen requirement, or both; 3, hospitalized, not requiring supplemental oxygen; 4, hospitalized, requiring supplemental oxygen; 5, hospitalized, requiring nasal high flow oxygen, non-invasive mechanical ventilation, or both; 6, hospitalized, requiring extracorporeal membrane oxygenation, invasive mechanical ventilation, or both; and 7, death. Time to recovery was defined as the first day during the 21 days of follow-up in which the patient reported that secondary outcome analysis. Secondary Outcomes included the proportion of patients with clinical deterioration, defined as those with worsening by 2 points from the baseline score on the 8 category ordinal scale since randomization. Sample size of 400 based on long rank test for the new primary endpoint was kept using an ivermectin to placebo assignment ratio of 1:1. The statistical methods used to compare groups for primary and secondary outcomes. So primary endpoint of time from randomization to complete resolution of symptoms with ivermectin versus placebo was assessed by a Kaplan-Meier plot and compared with a long rank test. So the results diagram is strongly recommended. So this is a diagram given. So for each group, the numbers of participants who were randomly assigned receive intended treatment and were analyzed for the primary outcome. Requirement dates defining the period of requirement and follow-up. Of the 476 patients who underwent randomization, 238 were assigned to receive ivermectin and 238 to receive placebo. 75 patients were randomized between September 2020 and were excluded from the primary analysis population but remained in the as treated population. 3 patients were excluded from all analysis because they were identified ineligible after randomization. The primary analysis population included 398 patients, 200 allocated to ivermectin and 198 to placebo. Baseline characteristics of the 75 patients who received ivermectin but were excluded from the primary analysis were not significantly different from 398 remaining patients in the cohort. So this is a table showing baseline demographic and clinical characteristics for each group. Outcomes and estimation. For each primary and secondary outcome, results group and the estimated effect size and its precision. Time to resolution of symptoms in patients assigned to ivermectin versus placebo was not significantly different. HR for resolution for symptoms were 1.07. And then in the ivermectin placebo group, symptoms resolved in 82% and 79% of patients respectively by day 21. Few patients had clinical deterioration of 2 or more points in the ordinal 8-point scale, and there was no significant difference between the two treatment groups. Then the harms, a total of 154 patients in the placebo group reported adverse events between randomization and day 21. 15 patients in the ivermectin group versus 5 patients in placebo group discontinued treatment due to adverse events. Serious adverse events developed in 4 patients, 2 in each group, but none were considered by the investigators to be related to the trial medication. And over here, interpretation. Cumulatively, the findings suggest that ivermectin does not significantly affect the course of early COVID-19, consistent with pharmacokinetic models showing that the plasma total and unbound ivermectin levels do not reach a concentration, resulting in 50% of viral inhibition, even for a dose level 10 times higher than the approved dose. Thank you.
Hello.
Hello. Yes, ma'am.
Yeah. Um, what conclusion do you derive from this study? Your
So,
understanding.
yeah, so my understanding, there was no much difference. And over here, among adults with mild COVID-19, a 5-day course of ivermectin was given compared to placebo. And there was no significant improvement for the time, like the resolution from the symptoms. So the findings do not support the use of ivermectin for treatment of mild COVID-19, although a larger trial may be needed to understand the effect of ivermectin on other relevant outcomes.
Okay. So, what were the Indian guidelines saying about how to use ivermectin during the first phase of COVID-19?
This is not approved yet because the clinical trial was done and this was a failure. And this was mainly carried out in Columbia. So
Uh,
the
yes, go ahead.
yeah, so they have now come to end the study because ivermectin there was no significant improvement at all. So the patient usually takes 10 to they have taken a time period of 10 days to see the improvement, and there was actually very slight improvement seen between ivermectin and the placebo they had given.
Yes, Joanne, if
So
I'm not
this
pronouncing it right.
yeah.
Uh, Indian guidelines has prescribed ivermectin as one of the most effective treatment during the first phase of COVID-19. So I'm asking, uh, what type of COVID for what it was used? Do you remember that? Are you aware of
Mm,
it?
oh no, ma'am, I was not aware of it.
Okay. Uh, One more question.
Okay,
So
ma'am.
initially, it's a simple question. Uh, Initially, ivermectin was used in what type of infections?
Over here was initially ivermectin is basically nothing but an anti-helmintic. So it was not basically used for viral infections.
Okay. If you are giving a patient counseling for a COVID patient in India,
Yes,
when a doctor prescribes ivermectin, what kind of patient counseling? What are the major points that you would counsel them with?
patient counseling for ivermectin.
Yes, not the general ones, specific ones related to ivermectin.
Okay. So this is basically an anti-parasite medication. So this might cause, especially over here in the adverse events, is given that it causes, or what to say, headache, severe headache. So that would be one of the patient counseling to make the patient aware that this is one of the side effects. And
Any,
there could be change in consciousness also.
Anything related to their lifestyle?
Lifestyle, benzodiazepines is usually not given with ivermectin. It's contraindicated when given ivermectin.
Okay.
Yeah. And they usually tell to take ivermectin on an empty stomach at
Okay.
least 1 hour or 2 hours after a meal.
Mm-hmm. Okay. Okay. So is this study generalizable?
According to me, I don't think so
Okay.
because there wasn't any much difference.
Okay.
And as stated in this article, the study was a failure.
Okay. What was the standard treatment given for the placebo group?
The standard treatment given for the placebo group is nothing but a mixture of 5% dextrose in saline and 5% dextrose in distilled water.
Okay.
After which, yeah,
So they have not used ivermectin as an adjunct, but they gave it as the whole treatment for the other cohort. Am I right?
yes, ma'am.
Okay. Okay. Thank you so much, Joan. Thank you.
Okay. Thank you.
We can move to the
Next,
next participant.
next participant is Trupti and Jeevita from Krupanidhi College of Pharmacy. Slot number SYCCON RXS48009.
Hello,
Hello,
Yes,
Trupti.
you're audible. Yeah.
is my slide is visible?
Yeah, it's visible.
Myself, Trupti from Krupanidhi College of Pharmacy.
And myself, Jivita R from Kripanadi College of Pharmacy.
So we are going to present a journal club presentation on the effect of low-dose corticosteroids and theophylline on the risk of acute exacerbation of COPD. The TAS-CS randomized control trial. TAS-CS is theophylline and steroids in COPD study. So this is the CONSORT 2010 checklist. It is consolidated standard of randomized trial. So there will be topic discussion on this and the checklist item and the page numbers in the articles present in this article. So background is, so there is highest burden of the COPD occurs in low and middle income countries. So low cost oral medication, if effective, could enable affordable, accessible COPD treatment for the low and middle income country. So the methods used are 3-arm, double blind, double dummy, placebo control study. They mainly conducted in the 37 centers in China. Symptomatic patients with moderate to severe COPD were randomized. They used 1:1 ratio to placebo twice daily plus placebo once daily, theophylline 100 mg twice daily and placebo once daily, and theophylline 100 mg twice daily and prednisone 5 mg once daily. So the mainly results, 1,670 subjects were randomized and 1,242 completed the study. So 1,142 with acceptable data at week 48. Subjects were mainly 75.7% were male and had a mean age of 64.4 years with standard mean standard deviation baseline force bronchodilator force expiratory volume in 1 second. It was mainly conducted by using St. George's Respiratory Questionnaire score 45.8 plus or minus 20.1. So conclusion, no low
Like,
dose theophylline
your slides are
alone.
not.
Can you see, ma'am?
Yeah, now it's okay.
So low dose theophylline alone or in combination with prednisone did not reduce exacerbation rate or clinically important secondary endpoints compared with placebo. So the mainly the guidelines used to follow COPD or Global Initiative for Chronic Obstructive Lung Disease, that is GOLD guidelines. According to that, the first line therapy will be short acting beta agonists and long acting bronchodilators. So theophylline is also a benefit. However, due to its potential toxicity when used in bronchodilating dose, it does not recommend as step of therapy for SABA unless inhaled long-acting bronchodilator are not available or affordable. So the main objective is we are going to compare the efficacy and safety of low-dose theophylline and oral prednisolone given daily for 48 weeks versus low-dose theophylline alone or placebo alone on time of first exacerbation and annualized rate of exacerbation in patients with moderate to severe COPD. Mainly it was 48 week randomized 3-arm double dummy study. So the mainly participants were male or female aged 40 to 80 weeks diagnosed with moderate to severe COPD, and we are going to use force vital capacity, and they had either a smoking history of the 10 years. It was mainly conducted in the June 2014 and May 2018. Participants were constantly recruited for symptomatic patients with moderate to severe COPD, conducted in 37 centers in China. That is the George Institute for Global Health, that is in Australia. So the primary outcome was the number of COPD exacerbation per patient in 48 weeks, analyzed as a rate prior patient per year. Outcome analysis, a subgroup of 75 patients based at 3 major centers had blood samples taken at baseline, 12 and 48 weeks for serum theophylline, which was analyzed blind in the central laboratory after completion of the study. Morning serum cortisol was measured in a subgroup of 91 patients at 50 weeks. The 50-week time point
2 weeks followed completion of the 48-week study. During administration was chosen to access adrenal recovery and the safety of administration of prednisone 5 mg daily for 48 weeks. So in this pilot study, we aim to enroll 2,325 patients, but 665 were not eligible to proceed to randomization at baseline. So participants were randomized 1:1:1 and randomization was stratified by patient diagnosis and recruitment site. Treatment randomization was stratified by smoking status and clinical site using a secure web-based randomization system and centrally administered in agreement with standard operating procedures on randomization at St. George Institute for Global Health. Study drug manufacture, packaging, and labeling was undertaken by approved Chinese suppliers and distributor. Research staff, clinician, and patients were blinded to treatment allocation throughout the entire study period. So it was mainly who blinded for the assignment to intervention was research staff, clinician, and patients were blinded to treatment allocation throughout the entire study period. The subject received either low dose corticosteroids or theophylline. To compare COPD rates, we fitted multivariable negative binomial regressions with a pre-specified covariate set with included treatment arm, hospital region, history of smoking, sex, exacerbation in the year prior to randomization, and post-bronchodilator, forced expiratory volume 1% at screening. For time to event outcome, we use proportional hazard Cox regression, and for continuous secondary outcome, where access the difference across treatment by ANOCOVA.
So the methods used for additional analysis were subgroup analysis, which includes age, sex, smoking, biomass, COPD exacerbation last 12 months, SGRQ score, that is St. George respiratory score. Results were the flowchart. Results were in flowchart. So totally, out of 2,325 participants, 635 excluded and 1,670 were randomized into 3 groups. So this double placebo, and another one is low dose theophylline plus placebo, and low dose theophylline plus prednisolone. Recruitment. So participant commenced enrolling the study in June 2014, and the last patient completed study on May 14, 2018. Altogether, 2,325 patients were screened and 1,670 were participated. Baseline data. So baseline data, the table shows which is the patients were randomized into based on placebo, theophylline plus placebo, and theophylline plus prednisolone, based on age, gender, BMI, smoking status, like that. Number analyzed. Participants were commenced enrolling the study in 2014, and the study was ended May 14, 2018. Outcomes. The primary outcome was based on there is no significant difference between the 3 treatment group for total, mild, moderate, or severe annualized exacerbation rates, whereas secondary outcomes were based on the forced expiratory volume in 1 second, SGRQ score, and CAT scores. Discussion. So there are 26% withdrawal rate in the study, with higher rate of withdrawals occurring earlier in the trial. Patient requested withdrawal if they felt the treatment was not helping them, and in particular if they suffered an exacerbation. Generalizability. So it indicates a treatment combination of low dose theophylline and prednisolone does not confer any benefits over the low dose theophylline alone, or low dose theophylline which didn't provide any clinical benefits compared to placebo. Interpretation. So there was no evidence of benefit of low dose theophylline plus prednisolone in the study. On average, the theophylline and steroid COPD study participants improved their COPD status during the study year. So other information. The protocol was assessed in clinicaltrials.gov. Funding. This work was supported by National Health and Medical Research Council, Australia, Stakey Laboratory of Respiratory Disease, and Guanlax Institute of Respiratory Disease, West China Hospital. Thank you.
Thank you, team. I would like to ask you some questions.
Yes, ma'am.
What is the immediate treatment given when somebody arrives at emergency with exacerbation of COPD?
Mostly oxygen.
Ma'am, mainly inhaled, what is that, bronchodilators, corticosteroids, that like
Budesonide.
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 Okay. What is the current guideline that has been used in South India or in general India?
Gold
Gold
guidelines.
guidelines.
Okay. What is the difference between an objective and an outcome?
Ma'am, objective, we are going to focus on what the things we are going to do, like we are going to find the efficacy or safety, like that. And outcomes will be if the treatment is effective or not, we are going to see whether the efficacy, safety, and these methods were followed properly and outcome was accurate to what the objectives we thought.
Okay. So you mentioned it is a double-blinded study. So
Uh,
what is the use of blinding? What bias, what type of bias is eliminated when you blind a study?
And we'll get to know whether the treatment blinding will be some it is some researchers will not know that whether treatment which treatment is given until the clinical trial is over.
To reduce some research bias like uh,
Yes. What type of bias is eliminated? Okay, this is a homework for you. You can go and look up to it. One more question. Um, this is a simple question. Which which enzyme or protein is deficient, which leads to the cause of COPD, and the causative factor is smoking?
Ma'am, can you repeat it?
There is an enzyme or a protein which is deficient due to smoking, which will lead to the cause of COPD. And what is that enzyme?
Ma'am, that is alpha 1 antitrypsin.
Okay. Thank you so much. We'll move to the next participant.
Thank you, ma'am.
The next participants are Shaila and Kalai Karuna Bhavani, JKK MMRF Annai JJK Sampurani Ammal College of Pharmacy, slot number SYCCON RX S4 80010. Okay. The next participant is Mallikarjuna Gowda, Sonia Education Trust College of Pharmacy, slot number SYCCON RX S4 80011.
Yes, ma'am. Am I audible, ma'am?
Yeah, you're audible. Can you please share your screen?
Yes, ma'am. Yes, ma'am. I am not unable to share the PPT, ma'am, because network issue is there, ma'am.
No problem. You can start presenting.
Yes, ma'am. Good evening, everyone. Today I am going to give a case journal club presentation on assessment of a drug utilization pattern in a chronic kidney diseases patients in a tertiary care hospital based on World Health Organization core drug use indicator. Introduction. A chronic kidney disease is characterized by a decrease in a glomerular filtration rate of less than 60 ml per minute, 1.73 meters square, 3 or more months, and abnormality in the kidney structure and its function. CKD was ranked in a 27th in number of deaths in 1990, and it is rise to 18th position in 2010. In India, the prevalence of CKD is 72.2%, and it was more in a male as a result of chronic alcoholism, diabetes mellitus, smoking, and hypertension and chronic vascular diseases. CKD is a significant contributor to comorbidities such as cardiovascular diseases and end-stage renal diseases. Such comorbidities can increase the treatment cost as well as the treatment challenges. The need for prescribing multiple comorbidities can increase the chance of developing adverse outcomes in a CKD patients. Uh, objective, the study the drug utilization pattern in a chronic kidney diseases by using the World Health Organization core drug use indicators. Drug utilization is defined as the marketing and distribution and prescription use of drugs in a society with a special emphasis on the resulting medical and social economic consequences. The study of utilization pattern in a specific setting helps in providing an idea about how the drugs are prescribed, which detect the presence of irrational use of medicines. And coming to the materials and methods, the study design and the study center. This is a perspective observational study conducted in an outpatient department of nephrology of Justice K.S. Hegde Charitable Hospital, Mangaluru, India. The duration 8 months from August 2018 to April 2019. Ethical approval. Study approval was taken from the Institutional Ethical Committee of Justice K.S. Hegde Charitable Hospital, Mangaluru, India. The samples size was taken to 56 patients. Inclusion criteria. The CKD patients above 18 years of either gender with a minimum 2 drugs per prescription. Exclusion criteria. The patient not willing to participate in the study. The data sources, the patient cases, drug prescription charts, and interview with the patient caretaker for demographic details. Materials used, the patient data collection form, informed consent forms, and World Health Organization core drug use indicators. The National List of Essential Medicines, SPSS 16.0 software. The World Health Organization core drug use indicators. The prescribing indicators. The average number of drugs per encounter is equal to total number of drugs divided by total number of the prescription. The percentage of a drug prescribed by generic name, number of a drug prescribed by generic name divided by total number of a drug prescribed into 100. The percentage of encounters with an antibiotic is equal to the number of prescription with
You can
antibiotic.
skip. You can skip that slide. You can go to the results directly.
Results, around 256 patients were taken the sample size in the stage 2. One patient having the stage 2 condition, and 9 patients have a stage 3 conditions. 37 patients have a stage 4. 86 patients having the stage 5 condition. 123 patients having the kidney failures. And distribution based on the comorbidities had at least one comorbid condition, that is hypertension. 230 patients have along CKD along with the hypertension, and 98 patients diabetes along with the CKD, and 65 patients anemia with an CKD, and 36 patients ischemic heart diseases, and 15 patients with along with the CKD and hypothyroidisms. And next, the drug utilization patterns. Out of 1,374 prescribed drugs, only in the 80.7% were prescribed by generic name, and 65% of the drugs were prescribed from the Indian National List of Essential Medicines, and total 256 prescriptions were analyzed with a total number of 1,374 drugs, out of which drugs acting on cardiovascular system were the most prescribed drugs. 586 drugs are the cardiovascular system drugs. And next chart, that is the drug usage pattern in a patient suffering from chronic kidney disease. That is the calcium channel blocker, the highest usage of the these drugs, calcium channel blocker, and lowest, that is the angiotensin converting enzyme inhibitor. 0.3% of the drugs can be used around these patients. And anemia patients, they have used the apotin alpha and intravenous iron sucrose and oral elemental iron and folate drugs. And gastrointestinal drugs, that is proton pump inhibitors and dopamine antagonist, H2 blockers and others. And vitamins and minerals, there is a 9.5% of the vitamins and minerals drugs are used in this case study. And calcium carbonate and vitamin D3 and vitamin D3 derivatives and vitamin B complex. Acid-base disorder drugs, the sodium bicarbonate and fepoxide, and anti-diabetic drugs, insulin and oral hypoglycemic agents, phosphate binders, that is a calcium acetate, and drugs for respiratory system, xanthine alkaloid and others, drugs for nervous system, antidepressant, anti-epileptic, antipsychotic.
You can skip to the discussion.
Discussion. Total number of 256 CKD patients were included in the present study, which revealed that the 75% of them were male patients. This is similar to the observation made by several other researchers. The higher incidence in the CKD observed in a male patients may be due to the role of sex hormones in the progression of the disease. On the other hand, the male tends to have a high protein and calories intake in the comparison with the females. Higher protein intake is associated with the initiation and progression of the kidney disease, and CKD patients with the multiple comorbidities may require polypharmacy. And 166 patients were prescribed with more than 5 drugs per prescription because of comorbid condition which indicate the polypharmacy. Calcium channel blocker 27.6% are the most prescribed antihypertensive, followed by diuretics, and centrally acting alpha 2 agonist 19.6% and 16.2% respectively. Conclusion. Displaying the national list of the essential medicines and circulating among the healthcare professional, increasing the use of drugs from national list of essential medicines which are considered more cost effective, and use of generic drugs to minimize the errors in the prescription writing and dispensing. Thank you, ma'am.
Okay, some quick questions. From the above table of the types of drugs that has been given, what are the drugs that should be completely avoided from the given table? What are the types of drugs that should be completely avoided in CKD?
Ma'am, most angiotensin receptor blocker, ma'am, because it can cause the most edemas in the bodies, ma'am. That's why the body does not, the kidney does not filter and removes the waste materials because of these drugs, ma'am, ARBs.
Okay, other drugs? What are the other drugs?
Other drugs, ma'am, that is AC inhibitor, ma'am. AC inhibitor, ma'am. Enalapril and captopril, this type of drugs, ma'am.
Okay. How do you measure the extent of damage caused by xenobiotics on the kidneys?
Can you repeat, ma'am?
How can you measure? You can specify a calculation or a diagnostic tool to
Yes,
measure
ma'am.
the extent of damage caused by a renal disease causing drug or a renal failure causing drug.
Renal causing drugs, ma'am. I didn't know, ma'am, this questions, ma'am.
Okay, okay. Thank you so much. We can move towards the next participant.
Thank you, ma'am.
The participants are over, ma'am. Thank you so much, ma'am, for your patience. And thank you all the participants for your active participation. Thank you so much, guys.
Thank you. Thank you so much. I appreciate everyone who has participated.
Thank you, ma'am.